Hi, I'm Ed Buckler. I'm a research geneticist with the USDA ARS, located on Cornell's campus in Ithaca, New York. Today I'm going to be talking about the past and future of plant breeding and how plant breeding can be used to tackle some of the challenges of climate change. Breeding is all about the design and selection of organisms for human purposes. We can look back over the last century to ask the question, what were the successes and weaknesses of breeding? One of the great successes was people like Norman Borlaug were able to improve the yields of both wheat and rice so that essentially a smaller proportion of the world's population had insufficient calories and protein. But they weren't able to necessarily give balanced diets to everyone around the world. Currently, we have too many people out there, the vast majority, who have imbalanced diets. A relatively modest number of the world's population has a healthy diet. What we want to do is really change these future curves. The vision we'd like to get to is a world in which we've changed those curves, where the number of people with insufficient calories or protein is pretty much eliminated through alternative proteins and adapted productive row crops, where we've been able to reduce the number of people with imbalanced diets with high quality fruits and vegetables being accessible and microbial foods. The next great challenge we have is we have climate change that's really starting to accelerate. We're going to have to deal with much higher temperatures, diseases and pests that are moving around the globe and moving into new environments, and we're going to have to deal with extreme weather, extreme droughts, extreme floods. And so how do we take on these types of challenges? The vision is by the end of this century, we've figured out ways to deal with greenhouse gas emissions, and we've actually gone negative by 2100, and we've adapted many of our plants to these new environments. Both of these visions, plants are really key. They're key to healthy diets, and because they're such an important part of the ecosystem, they're really key for anything we do with greenhouse gas emissions because they occupy over 80% of the globe's biomass. Plant success is really about the plants being adapted to their local environments. And plants have adapted to thousands of environments all across the planet, given millions of years. However, with the help of humans, they've also been adapted to our fields or our greenhouses. And so as we look towards the next century, the question is, how do we get all these plants adapted to the new environments that they're going to be encountering? After a century of science, breeding now has an amazing set of tools. And I'll break down those tools into what we call breeding 1 through breeding 4. Breeding 1 is really about tapping diversity and using phenotypic selection. Breeding 1 really starts as 300,000 different plant species adapted to environments all across the planet. But then 10,000 years ago, humans started domesticating those crops. And so now about 2,500 species have been domesticated. And every country relies on crops that have been developed and domesticated in other regions of the world. In the US, the USDA National Plant Germplasm System is responsible for preserving and sharing this incredible diversity. Overall, they maintain and study about 16,000 different species that include nearly 600,000 different varieties of crops and crop relatives. But how do we make sense of this incredible diversity? In collaboration with CIMMYT, the leading institute for maize and wheat breeding for the developing world, we worked on approaches that combine genomics with environmental adaptations to identify some of the variation responsible for those adaptations. It turns out that thousands of genes are responsible for maize's adaptation to both North and South American environments. The key point to realize is there is not a small number of wonder genes. But if we're really going to tap the full diversity that's out there, we need to also learn from wild species. Recently, with colleagues, we've begun developing approaches to identify the source of adaptations across species. In this case, we're looking at 1,000 species closely related to maize and sorghum that have adapted to extreme temperatures, drought, and flooding. And so, as we close out this area, one of the big challenges we have in breeding, one, is really indexing the library of life. Biology and agriculture over the next decade really need to sequence, for example, maybe the million key plant, animal, and fungal species, and about 1 million key varieties of crops. Those seem like really big numbers, but for context, uh, by middle of this decade, we'll probably be sequencing about 50 million genomes per year for various medical purposes. Earth Biogenome Project is an effort that's just getting underway to do exactly that. Try to sequence about 1 million key species in the next 10 years. So once we've had that index, though, we really need to start deploying diversity. And I think the USDA's GEM program, the Germplasm Enhancement Project with maize, is a great example of that. It's a public-private partnership. Seed companies are providing elite inbreds for crossing with these diverse materials, and it allows breeders to explore diversity in a pre-competitive environment. A little over 100 years ago, breeding 2 starts. It's really centered around controlled genetics. In this phase of breeding with controlled genetics, breeders, for the first time, really knew the pedigree of all their varieties, and they could create genetic clones at scale. This provided insights into key phenomena such as heterosis or hybrid vigor. But also, at this period, there were key tools that were built, such as research harvesters, were really important for measuring performance and creating crops that performed together. And at the same time, the fields of statistics and experimental design were substantially born out of the need to make sense of this agricultural data and just pick out the winners and understand how we're interacting with the environment. The big challenge that we have today is how do we relate information at the DNA level to those interactions with the environment? The Maize Genomes to Field project is doing exactly that, with 30 groups working together every year to put together corn varieties all across the country. Over 200 different combinations of environments have been evaluated, and these studies are really critical for expanding our knowledge of how breeding can adapt plants to climate change. The extreme events of today might be the commonplace of tomorrow, and by studying such a diverse set of environments, we can prepare for it. Finally, we're really bringing new eyes to the field. Autonomous technology and vision are allowing us to look at plants in totally different ways. Rovers are allowing us to explore underneath the canopy and drones from above. But essentially what we're able to now do is record growth dynamically through the season. And most importantly, we're learning to record and understand how plants are reacting to their recent environment. How did a plant grow over the last week when there was a lot of rain or when there's a lot of heat? Breeding 3 is about using genetic markers. Genetic markers are ways that we can track what's going on in the genome. Starting about 40 years ago, we were able to do very basic, what we call marker-assisted breeding. Follow and track a few genes, a few loci across the genome. Today, though, we can track the entire genome, and we have the mathematical tools to make predictions about how the whole genome will perform. The breeding cycle is the key element for getting genetic gain in a population and producing long-term gains in breeding. The process works by selecting parents, crossing them, and then evaluating their offspring for everything from disease, how they grow together in terms of yield, and what product they produce. It works. Eightfold gains in the case of maize were achieved over a century, but it takes time. The problem is that evaluation process can take up to 5 to 10 years. This is where genomic prediction comes in. If you have good data to predict, these advancements can happen almost as fast as you can make offspring. Theoretically, 15 times faster. Reality is not quite like this, but substantial increases are possible. We've already seen genomic prediction and genomic selection deployed to a couple species. For example, in cattle, the USDA organizes national prediction models that are used for cattle breeding. In the case of maize, major seed companies use this regularly to advance their varieties. The challenge we really face is now deploying these types of approaches for genomic selection to all species. Our challenge is here that genomic prediction requires accurate mathematical models. The way we develop those for major crops is we collect lots of observations, and we can then relate an accurate model between the genotype and the phenotype. And these have high accuracy. However, if we're working with a specialty species, there may be relatively few observations, and we end up with low accuracy. So the challenge is, how do we increase that accuracy? And this is where we're trying to figure out if we can add information in from all these other areas of science, whether molecular biology, biochemistry, evolution, what we've learned in other species, to get back to that high accuracy. Breeding 4 is about molecular design. This is the situation where we've identified the genetic variant or the gene that creates a particular trait. And starting in the 1990s, we were able to use this to create GM crops. And now more recently, we have technology to do editing. And you'll hear a lot more about that from Zach Lippman. I'll just mention a couple examples where GM was been deployed very successfully. For example, in papaya ring spot virus, Dr. Gonzalez's team developed a GM solution that's really been effective and durable and allowed the Hawaiian papaya industry to continue. In the case of maize, I worked on an area where we were trying to improve levels of provitamin A to improve nutritional status of people in sub-Saharan Africa. And using natural variation, we were able to increase the amount of provitamin A by over 20-fold. However, scientists working in rice didn't have access to the natural variation maize does. So there they used actually some of the exact same genes we have in maize, and they inserted them into rice, and were able to make varieties of rice, now we call golden rice, with high levels of vitamin A, just like maize has. But our real challenge going forward into the future, especially when it comes to climate change, is we know that thousands of genes are involved in adaptation and yield and nutrition. But which of the variants matter? If we look at two varieties of maize, they may differ from one another by millions of genetic variants. And across the species, there may be 80 million genetic variants. How do we go from those 80 million genetic variants down to the few thousand or 100,000 important variants that we want to use for adaptation? Just as we discussed with machine learning, we need to start applying knowledge from all the other areas of biology to help us filter these 80 million variants down to the 100,000 that matter for yield, adaptation, and nutrition. So with all these incredible tools, how do we apply them to adapt our crops and combat climate change? I think these tools can be applied towards 5 major targets. First, apply them to numerous species. Second, fight disease. Third, adapt them to climate change. Create circular agricultures. And finally, sequester carbon. Let's first talk about applying them to over 1,000 species. Why is accelerating breeding for 1,000 species important? Well, most advanced breeding to now has just focused on about 10 species, mostly focused on yield. But if we're going to go after all these other targets, such as a circular agriculture, nutritious fruits and vegetables, fuels and polymers, carbon capture, and assisted migration, we're going to start needing to accelerate breeding for thousands of species. One route that's really important for accelerating the breeding of many of these crops is putting the tools, the information tools and the genomic tools into the hands of breeders that are working with these crops and making them easy to use so they can deploy them without having large teams. And that's what USDA ARS's Breeding Insight is doing, is building those systems to allow a much wider range of breeders to be able to use these advanced tools. One of our challenges, though, remain is how to efficiently apply the rules of life to breeding. And as we keep on learning these rules over the next decades, it's then coming up with the information systems that allow us to really apply that knowledge to the GS process, whether it's in using the natural variation that's already out there or nominating hundreds or thousands of edits to go into CRISPR. But I think we're getting to the point in the next couple of decades where we're going to be able to do this and apply them to a much wider range of species. And I think you'll hear from Zach Lippman some of those ideas. The next area we're going to talk about is fighting disease. Our biggest challenge with the disease is we don't know exactly what will happen. But we can be sure if we change global environments, then new diseases and pests will be on the move. And our biggest issue is how do we rapidly respond to these? In the early 1900s, wheat was facing big problems with stem rust. And it wasn't until about the 1960s that the deployment of single resistance genes really started addressing the issue. But then in the 1990s, UG99 cropped up, and there was essentially no resistance among U.S. varieties against it. And so the USDA ARS, combined with researchers from around the globe, went to go identify more sources of resistance. And working with wild relatives, and they identified 10 alien genes, cloned multiple genes responsible for resistance, and then we're able to use these marker-assisted approaches to quickly stack the genes together and have now produced highly resistant varieties. And this approach is only getting faster as they are now being able to deploy genomic selection. The same type of pattern has happened with wheat and barley fusarium head blight. For about 100 years, there's sporadic losses, but then it evolved, and by the mid-1980s, there were heavy losses began. And again, people were able to identify key resistance loci, use genetic markers to provide resistance, and stack those together. And that type of approach of identifying the genes is now getting a lot faster with the sequencing of the wheat genome and the tools of genomic selection becoming available to breeders all across the US. However, we've not been so successful in the case of many trees. For example, we lost the American chestnut from the eastern United States, and the bark beetle is now killing conifers across the West. There are approaches, for example, speed breeding combined with genomic selection or genetic modification could help rescue some of these trees. But we need to come up with a wider range of approaches for these perennial species. And while plant breeding can be a really powerful tool for addressing disease resistance, sometimes it may be just too slow. And the example here is for citrus greening. It's swept in, destroyed much of the Florida citrus crop. In this type of situation, we really need to develop biological therapies that are combining novel delivery approaches with molecular medicine, combined with the power of plant breeding. So overall, when we think about plant diseases and pests, we need to be fast, flexible, and global. Surveillance needs to be global. Resistance needs to be discovered in crops and non-crops. And then deployment needs to include approaches like genomic selection, genetic modification, genetic editing, and a wide range of molecular therapies. And instead of taking 20 years to deliver these, we need to figure out approaches to deliver them in 5 years and deploy them. So how do we use these great tools to adapt our crops to future environments? So the first thing to remember about adaptation is water availability is central to crop yield. And water availability is tightly tied to the amount of rainfall and the amount of heat. And over the next century, in places like Iowa, we're looking to see much higher temperatures. And at the same time, we're likely to have extreme weather events where we both have extreme drought and extreme flooding. And so how our crops can deal with these two things, it will be really central to our ability to maintain crop productivity. To deal with this, we need to use a wide range of strategies. One is we need to breed for temperature tolerance, for essentially those extreme temperatures. We need to breed so that the proteins are stable in these plants, and that's likely to require modifying and selecting on thousands of proteins across the genome. We may need to modify these crops to allow them to access water in different and better ways, for example, changing their roots and increasing irrigation. And then we're likely to need to modify some of the approaches they're doing photosynthesis, which is when they lose most of their water. That may be adding pathways from species that have been adapted to high temperatures. But in some of these situations, we may just need to avoid the summer heat, just as winter wheat and winter barley do. So you could imagine us actually changing crops such as maize and soybean to becoming overwintering, where we have a fall planting and they essentially grow throughout the whole year, and we harvest them at the end of the next summer. But there also are going to likely be many crops we really can't adapt to these extreme temperatures. And those are crops that are likely need to be moved into greenhouses and shade houses, where we can make efficient use of nutrients and water, we can reduce transportation costs, and if we put them under sterile conditions, they will have a relatively low disease pressure. Our next major target is circular agriculture. Just as water is a key determinant for yield, so is nitrogen. Circular nutrient economies are about reducing the amount of waste nitrogen and phosphorus. Nitrogen and phosphorus inputs are really key to producing high yields, and they also then produce high levels of starch, food, and feed. But if we can reduce that waste nitrogen and phosphorus and reuse that on the farm, we end up with a much more circular nutrient economy. So the real question is how to keep more of that nitrogen and phosphorus on farm. And when you look at crops like maize, you see that they're very efficient in terms of energy. Energy in versus energy out looks great. But it could be far better if we were able to reduce the amount of energy coming in from our nitrogen fertilizers. And if you look at greenhouse gas emissions, you realize that the nitrogen fertilizer and nitrous oxide releases off of our maize fields are the main source of the greenhouse gas emissions. And the final problem with nitrogen and phosphorus is that they run off our fields and end up in places like the Gulf of Mexico, where 60% of the nitrogen is coming from agriculture. This is a loss of nutrients, which is an economic loss to farmers and an environmental cost globally and nationally. Fundamentally, this is why we need to start breeding for an entire agroecosystem that include things like cover crops and perennials. Integrated breeding and management can improve soil biology, nutrient cycling, drought, and flood tolerance. But I want to also remind people there may be other strategies we can use. Fall colors are a reminder and a sign that perennials are recycling nitrogen and phosphorus. Are there some strategies we can borrow from these types of plants that we could use in modern agriculture? Fundamentally, could we make annuals sometimes more like perennials? A graduate student working with my group suggested the concept of why don't we store excess nitrogen and phosphorus in the cob and save that for the next year's crop? This may work, it may not, but this is the type of idea and approach we need to try for the future. Get to our final area. How can we use these new tools to actually combat climate change and sequester carbon? Circular agriculture is one of the key ways we could do that by reducing greenhouse gases. Of course, we need to reduce the greenhouse gas emissions coming from our energy production systems, but things such as fertilizer production require about 1% of the globe's energy, and reducing their use could reduce energy usage. Nitrous oxide emissions, as we've mentioned, are a major greenhouse contributor, and we can have targets to reduce those, both of which are really result in greater efficiency as a double win. The next area is sequestering carbon. By the end of the century, we need to be actively sequestering nearly 20 gigatons of CO2 per year. And plant breeding and plants in general are one of the key ways that one could sequester carbon. I think there's at least 3 different approaches we need to think about where plants can play an important role and plant breeding can play an important role. The first is in fuels. We're already seeing this with bioethanol. Can essentially replace gas. And it can be efficient, especially if it's refined with solar or wind energy. Crops could also be used as the base polymer for plastics. Again, if solar and wind energy is used to process them into the polymers and plastics we need. And then finally, I think there's a lot of opportunity for using the photosynthetic capacity of our crops and our rangelands to produce deep carbon for sequestration. So there are some strategies that allow us to really sequester carbon. Well, perennials actually already sequester some carbon, but not very rapidly and not really enough. My colleague here, Jerry Glover, shows an example of some of those perennial grasses growing, and most of the carbon they are capturing stays above ground or in the top portion of the roots. But could we change that? Could we start really breeding for deep, persistent carbon? And I think we certainly could. We could probably start moving much more of that carbon down to the bottom and releasing it there where it's much less likely to break down and come back into the atmosphere. And we could probably do that for both our perennials and our annuals. And if it's accompanied by a lot of the other optimizations and technological improvements we have, the harvest loss above ground may not be that great, and we may end up with greater resilience. Using this type of strategy, if some of future yield gains went to carbon sequestration, U.S. lands could sequester nearly 3 gigatons of CO2 per year. That's about 14% of the global target. And if you look at what that would be economically worth, at estimates of maybe $50 per ton of CO2, that would make those U.S. lands' carbon sequestration value at about $42 billion per year. If we want to make 21st century breeding success look like this, essentially healthy diets around the globe and going carbon negative by the end of the century, then we really need to think about how we're going to take these incredibly powerful tools we have for breeding and apply them systematically to increasing their application for 1,000 or more species, fighting disease, developing a circular agriculture, adapting our plants to climate change, and sequestering carbon where possible.